Okay. Good morning. Yeah. So I hope you guys did well yesterday. Many of the members didn't post marks. So, okay. Take time, correct, and if you have doubts, any key doubts and all, please ask me and please post the marks. Okay. So today we're going to discuss about, one of the plant physiology, lesson, that is photosynthesis. If you see the weightage of this chapter, it's almost 3.5 or 4% weightage is there, right? Out of 360, out of 100, you have got 4% chances that you will get questions from this, right? So especially when physiology is concerned, animal physiology is also more important than plant physiology. But within the 11th standard, 22 chapters, this both plant and animal physiology will have more weightage, right? Good morning, Akshaya. Good morning, Abhiniti, Vishalini, Shamili, Arshia, and Jeena Priya. Kanishka, right? Yeah. Good morning, all. So let's start with begin with. So photosynthesis chapter, it's a very vast chapter actually, because we have to we'll be touching different types of reactions in this, right? So what are the different reactions, and what are the light reaction, dark reactions, right? How come the plant preparation food material is really not depend, not really doesn't matter. Even nighttime also it takes place, in the sense like we usually think that the light is during the daytime only. There's all this process, it means the preparation of food may take place, but actually if you see, there is a light reaction and dark reaction, right? So the light reaction products will be utilized for the dark reaction. So what you have to focus here is, right, the products of light reaction, and how many molecules it produces per CO2, because it is fixing the CO2 molecules, right? So it's fixing the CO2 molecules. So you should be having this how many molecules of CO2 are utilized or fixed to form glucose. At the same time, other other at the end of to happen. So across the kingdom, we will be talking about more autotrophic mode of nutrition, where you can see that especially these photosynthetic organisms are able to utilize the sunlight and the pigment that has, right? By presence of the sunlight and the pigment, they will be fixing the CO2 into glucose molecules. So glucose is an organic compound. So 6 CO2 molecules, right, would eventually give rise to one glucose molecule. So this balancing of equation also very much important, right? Right. So if you see, the entire process needs to be demonstrated. We can demonstrate that this entire process can be of light dependent and light independent. So far what we are seeing is that right now there are photosynthesis taking place in a number of plants around you, where the light reaction is taking place. So the reactions that occur by the presence of light, or by depending upon the light, by using the pigments like chlorophyll, right, in the chloroplast, those products like NADPH2 and ATP, right, they will be produced. And these are the currencies which will be utilized for actually preparing the starch, which is a light independent reactions. So these are the two phases you need to demarcate. So whenever you think of photosynthesis, in your mind you should get two separate pathways. One is light reaction and the dark reaction. So light reaction means it's involving in light. So what are the different processes we can really see? Oxidation, reduction, whether the energy is consumed or whether the energy molecules are formed. If they are consumed, how they are consumed, how many molecules of ATPs are utilized. So these are the important aspects you need to focus, right? Is my voice everything audible and no issue, right? So, so when you try to see that what are the important aspects we need to cover in the light reaction. So I'm again saying this is not a formal class that we'll be talking this photosynthesis deeply. This is just an outline and important highlights, right? If we had to take deep discussion, it will take almost a weekday to complete this chapter itself. Okay, because reactions are involved, and you will be finding different proteins and channels will be involved, right? For example, cytochrome, proton acceptors, electron acceptors. We will see those things, right? So let's move on to the actual aspect. So when you see this, you should look this photosynthesis into two phases: light dependent and light independent. Okay, so when it comes to photosynthesis, you should be focusing on the scientist name actually. In the sense, like we have few scientists which they talked about, right? So Hatch and Slack. So Hatch and Slack, there is a pathway, right? And there is a Melvin Kelvin pathway. In Melvin Kelvin, they got Nobel Prize also. So don't ignore this scientist name when it comes to this photosynthesis, right? And for example, dark reaction, Blackman. So these are the different few scientist name you need to remember. At least like when you are reading the pathway itself, you can actually practice those names. Okay. So cell organelle is concerned in the photosynthesis is a chloroplast, and you know the structure of the chloroplast where it is a double membranous structure, right? So it will be having outer membrane and inner membrane, and you will be having thylakoids membranes, like building story buildings, right? 10-story building or 15-story building, like an apartment. You'll be finding thylakoids placed stacked over one another. So if you see here, so if you'll see that these thylakoids are stacked over one another, like this, stacks 1, 2, 3, 4, one on the other, and the thylakoids again connected by stroma lamellae to other stack. It means that imagine that your classmates or your friends are staying in a gated colony. It's a big apartments. So for one apartment to the other apartment, there is an interconnecting bridge, that is stroma lamellae, right? So why I am explaining this stroma lamellae? Because this PS1 is present in this stroma lamellae. Okay. So you will be having these stacks, and there will be many stacks like this within the chloroplast. Assume that this membrane, inner membrane and outer membrane. Outer membrane is like a fencing, and inner membrane is like your some what do you say, security guards are there which guarding these apartments, right? So why these are important? Because where exactly this location of photosynthesis and the structures of this would be always confusing actually. So whether it is occurring in thylakoid or stroma or lumen where exactly present, those are these granum. So these are the different terminology, you will get confused. So these are the thylakoid membranes and the stacked, these are thylakoids, and it will have membrane, thylakoid membrane, and these entire stacks grouply we call it as granum or grana. Okay. So they are all no. So within this thylakoid, you will be finding lumen. Okay. So remember that when you open the book, you will be finding this is a lumen. Okay. So why lumen is important? Lumen, where the place of H plus ions accumulation, which leads to proton gradient ATP synthesis. It means this thylakoid is there, right? So this thylakoid, these are stacks of thylakoid, and they are interconnected, and there are other thylakoids, and within this thylakoid has a membrane. At the same time, it has lumen. So within the lumen, what happens is hydrolysis takes place, splitting of water molecules, and creates protons. So these protons, when they are pumped out, that's what ATP synthase. You would have remembered ATP synthase. What do you mean by ATP synthase? What is the function of ATP synthase for that matter? Which is the smallest motor in the universe? You would have known last year itself. What is the function of ADP synthase? What are the components of ADP synthase? Where exactly the location of ADP synthase? So these aspects we will try to address. Okay. So within this chloroplast, these are the different structures are extremely important. It means stroma is there, right? The stroma, within the stroma, you will find the thylakoid stacks or arranged like granum. And within the thylakoid, you will be having lumen, where the proton gradient will be produced. And this thylakoid will be thylakoid. You will find PS2. PS2. PS means here photosystems. You have PS1 and PS2, right? So PS1 is situated in the stroma lamellae, and the PS2 is situated within the thylakoid. So within this thylakoid, you will be finding that, because the presence of pigments, the light reactions would be taking place. Okay, more than this, the chloroplast structure has no that's enough. You can cover this structure in the cell structure chapter. Okay, so this is the organelle, cell organelle, which is responsible for the photosynthesis. You have got no answer. What is the ATP synthase or universal? In the universe, this is the smallest motor, right? So if you see that the smallest motor, so you can imagine the smallest motor would have two subunits, okay, like this. Okay, two subunits like this in the membrane. So this is a membrane, right? So when you see this membrane, right? Yeah. So within the lumen, right? And you have got thylakoid membrane, right? So it would be present. It would present like this. Imagine that my hand is a membrane, thylakoid membrane, right? So it would be like this. So intermembrane, just like integral proteins, they will be present embedded within the membrane like this. So what happens is in the lumen, this protons would produce, and this F1 and F0 particles would actually rotate like this. When they're rotating now, what happens is the protons would come out, and finally that ADP would be converted into ATP. So this is a smallest motor. At the same time, this actually, this function, this ATP synthesis function is to convert ADP into ATP. So likewise, the energy would be stored in the form of ATP, right? So that is what important aspect that you need. ATP synthase function and structure is extremely important. Okay, don't ignore that. And so let's move further. So when comes to chloroplast structure, these are the things you need to focus. That is location of PS1, location of PS2. So what do you need to focus? Location of PS1 and PS2. Focus on this. Okay. ATP synthase ATP from ADP to inorganic. Yeah, correct. Yeah. So ATP synthase enzyme is integrated protein motor, right? So F0 F1 particles would rotate, right? So that it actually converts ADP into ATP. That is one of the important aspects. Yeah, I'll try to show on video. Okay, I'll send that. Just try to see that. After few minutes, I'll send that when we are taking that aspect. Okay. Fine. So let's move on. So when we talk about the pigments, so pigments are the essential components that actually trap this sunlight, right? And if you see this plant pigments, there are two types. One is photosynthetic pigments and non-photosynthetic pigments. So when we talk about photosynthetic pigments, there are fat soluble pigments and water soluble pigments. Okay, so you would have heard about different pigments. So this is a classification, and this type of classification may come in section A and section B. If you have the NCERT textbook with you, you better keep have an access to that also. That would be easy. So when you see the graphs that were given in NCERT, those graphs, absorption spectra of this chlorophyll A, B, C, D, E, these are all very much important, the absorption spectra. So they will be giving different graphs, and they may ask you which absorption spectra belongs to which chlorophyll. Okay. So within the fat soluble pigments, we have different chlorophylls. That is, they are green colored one, especially the chlorophyll A, B, C, D, E, right? And then we have carotenoids. Carotenoids, we have carotins and the xanthophylls, right? These molecular formulas would vary, but molecular formula, you don't need to really, just have a look at it, but you don't need to really mark anything about that. So chlorophyll A, B, C, D, E, and those are all green colored, and you have a carotenoids, which gives yellow color, especially the carotins. Yellow color will be given, and then there are xanthophylls, which give brown color. So the color and the name of the pigment, and whether soluble in water or fat, and whether they are photosynthetic or non-photosynthetic. This is what you have to have an approach. When it comes to non-photosynthetic pigments, you have anthocyanin, anthocyanin, right? So purple, green, purple, red, blue, green, and these are the different colors that you see, which are non-photosynthetic pigments, right? Red, purple, blue, green. Like anthocyanin is a red color giving pigment, etc. So pigments you need to focus. And what is a universal photosynthetic pigment? If you see on by and large, you will find you try to isolate this chlorophylls. You will by the thin layer chromatography technique. We can by chromatographic techniques we can really isolate, and we can study this chlorophylls also pigments, right? If you see that chlorophyll A and beta carotene would be the most abundant, or in the sense like universal photosynthetic pigments. Okay. So when we talk about the mechanism, so these are the few. Coming to few more facts. The heaviest pigment. Now, if you see the molecular structure, right? So you have the heaviest pigment, that would be chlorophyll B. Okay. So chlorophyll B is an heaviest pigment. When you compare this chlorophyll A, B, C, D, their molecular formula and structure, you will be identifying there is a difference in the oxygen molecules, okay, oxygen atoms. So this makes chlorophyll B heaviest, and the lightest would be carotene. Okay, So these things you need to just brush up. Okay, which is the heaviest chlorophyll and which is the lightest no pigment. Okay, and coming to the next one, mechanism of photosynthesis, where you need to focus on the stages. So there are this. I already told this photosynthesis can be divided into two phases. One is photochemical. It means this chemical reaction, this entire reaction, which are driven by enzymes, which is taken in the presence of light. So photochemical phases. And the next one is biosynthetic phase. Right. So let's see what is a photosynthetic phase or light reaction or Hill reaction. Okay, when they are asking these synonyms, you need to be extremely careful. Okay, so in this Hill reaction or light reaction or photochemical reaction, you'll be having the end products, right? You know, they produce this ATP and NADPH molecules, and this ATP and NADPH molecules are very, very, very important for the next reactions to take place. In the sense, next phase, that is biosynthetic phase, synthetic. So see, in the name itself is a synthetic. What is this actually synthesizing is nothing but glucose. So glucose is it's an reduction product, right? Isn't it? So the CO2, there are different enzymes involved, or Rubisco is involved. There are different steps we'll try to address. So it is reduced and it is fixed. CO2 is fixed to form this glucose molecule, and it would be the reaction would be opposite when we talk about respiration. The same glucose molecule would be broken down into simpler inorganic molecules, right? So catalysis, right? So oxidation would be taking place, whether it is in the presence of oxygen or absence of oxygen makes it different respirations, right? That is complete, incomplete, like fermentation, etc. So let's see what are the different stages that includes in the photochemical phase or light reaction or Hill reaction. So these are the following events that occurs during this Hill reaction, where you need to more focus on the light absorption, in the sense like which photosystem is absorbing. For example, PS1 and PS2 are there. There are two photosynthesis are there, right? So within this photosystem 1, the reaction center is the pigment system P700. Here P700 is a wavelength, 700 is a wavelength. So at that wavelength, this absorption would be taking place, right? So the light which has that absorption, that would be, this light would be absorbed at that particular wavelength. So you need to focus on those wavelengths. Okay, are you guys following? Are you guys following? I'm just giving the highlights, like which you need to focus actually. Okay. Okay, fine. So in first aspect, so within this light reaction or Hill reaction or photochemical reaction, you have 4 different types of events that occur. One is light absorption, splitting of water molecule, that is hydrolysis, release of oxygen, and formation of high energy chemical intermediates. So these are the 4 steps involved in the photochemical reaction or light reaction or Hill reaction. So when you talk about the light absorption, you have certain components that are actually really necessary for to reactions to occur. So the molecules which are responsible for the absorption of this light is a protein based complex, that is light harvesting complex, LHC, light harvesting complex. So it is harvesting, it is absorbing, the light is nothing but the quantum of energy, right? This packets, right? So packets of energy, right? So this this light energy will be trapped or harvested by this light harvesting complex, which is organized into 2 photosystems. One is photosystem 1 and photosystem 2. And right now we know that where exactly photosystem 1 is there, photosystem 2 is there, right? So when we talk about photosystem 1, the reaction center in this pigment system is 700, P700. It means which absorbs the light of the wavelength 750 nanometers. So there is, when we talk about the light, right? Light and light in it, we will be you will be studying in even physics also. Different wavelengths, right? So visible, and you have UV, you have different infrared, IR. There are different wavelengths. So where exactly which wavelength actually the plant would utilize? That is the focus you need to have, and those are all given in NCERT very beautifully. The graphs you shouldn't ignore. Okay, so this 700 nanometers would be absorbed, and it has more of chlorophyll A, chlorophyll B, and carotenoids, right? So compared to other chlorophylls, it will have A, B, and you would have seen this NCERT. They have very clearly mentioned that chlorophyll A has this much absorption. So this, this absorption wavelength. Chlorophyll B has this called absorption wavelength, right? So those wavelengths you need to focus. Okay, so which chlorophyll absorbs in what absorbs which wavelength of the light, right? So which has maximum which has maximum absorption phenomena, which shows that maximum absorption also you need to focus, especially means that those graphs, right? So that green, yellow, blue, the graphs which were given, right? So ABC graphs which were given in NCERT, you should focus on them, right? Suddenly they give the graph and they'll remove this chlorophyll A, carotenoids, chlorophyll B, and they might be asking that which has the maximum absorption, right? So that you need to focus. Yeah. So this is about the photosystem one, right? It will be having more of chlorophyll A, B, and carotenoids, right? And the PS1 can carry on cyclic photophosphorylation independently, right? So here we have a different scheme of electrons would move from this photosystem 1 to photosystem 2, or photosystem 2 to photosystem 1, right? And they enter into electron transport chain. So there is a movement of this electron. So there would be jet scheme of movement also we'll try to talk about. So when we talk about photophosphorylation, so phosphorylation in the presence of light, so the PS1 can carry cyclic photophosphorylation, and you have photophosphorylation two types, one is cyclic and non-cyclic. So PS1 can carry cyclic photophosphorylation independently. So, and what else? The PS1 with electron carrier is located on both the parts of grana thylakoid and stroma thylakoids. So there are grana and stroma thylakoids where this PS1 is located. When it comes to photosystem 2 or pigment system 2, that is P680. So functions as a reaction center in this photosystem, and the photons of the lower wavelength are absorbed by this photon. So when compared to 700, you have got 680 is a lower wavelength. So the lower wavelength of the light would be absorbed by this photosystem, right? It is located in the parts of the thylakoid and carries out non-cyclic photophosphorylation with the PS1. So non-cyclic photophosphorylation takes place. So when we talk about photophosphorylation, then it will be clear, right? So what are the pigments that you see in this photosystem? So in this photosystem 2, right, has chlorophyll A, B, and carotenoids, almost the same as well as xanthophyll, where the xanthophylls also function as antenna in this system. So they will be trapping the sunlight. So these are the 4 different pigments that you can see in photosystem 2. And then next, splitting of water, the second aspect. First aspect is the photosystems, and then next aspect is photolysis of water or splitting of water. So during this photosynthesis, what happens is the water is used as a source of hydrogen, right? The protons required for the reduction of CO2. Reduction in the sense like, what do you say? Gain of electrons, GER, right? Gain of electrons is called reduction, right? So this CO2 is reduced to form this in presence of light and chlorophyll, they are forming actually glucose. So during this process, these protons, which are formed by splitting of water molecules, which is within the lumen, right? This this or outside the lumen, where you will find the splitting of water molecule would eventually donates the protons for the reduction of CO2 to form carbohydrates, right? Van Niel and R. Hill. So these are the two scientists who actually demonstrated for the first time this water splitting, photolysis of water. And the next one is high energy chemical intermediates like NADPH2, NADPH, and ATP are produced during this light reactions. So within the formation, how this higher energy molecules are formed. So they can form by photophosphorylation, where we have two types. One is cyclic and non-cyclic, and we have chemoosmosis in chloroplast, chemoosmotic theory, right? So chemoosmosis, chemo is the chemical, osmosis means there is a gradient, right? We would have studied in transportation, right, yesterday. So there would be a gradient of some molecules. Here that gradient is nothing but protons. So by the creation of gradient, chemical gradient, that because of the proton, how we can really produce this ATPs and NADPHs, right? That is what the formation of higher energy chemical intermediates, that is where you need to more focus. Photophosphorylation, non cyclic and non-cyclic, and what is the schematic representation. Okay. Fine. So when we talk about so the photons, once they hit this photosystems, light harvesting systems, then there will be a primary acceptors, and there will be some reaction centers, and then they actually transfer these electrons to the primary acceptor center. That's what they have shown in the light harvesting complex. And then when we talk about the electron transport, we have a jet scheme of the electron transport, where the electron transport, the electrons are transported from one one electron acceptor to the other electron acceptor in a jet scheme. Okay, jet scheme would be taking place where you can see that this NADPH were utilized during this jet scheme of light reaction. So that jet scheme you need to focus where the light hits the light harvesting complex. I mean the photosystem. You have pigments, right? It would be harvested, and eventually the electrons would be sent to photosystem 2, right? And that from the photosystem 2, right, which from the photosystem 2, what happens is this electron enters into the electron transport system. Again, from those electron transport system, this electrons would come to the photosystem 1. So this is a scheme of the electron transport in the light reaction. So that is a jet scheme of light reaction. So once you just go through, so where first is photosystem 2 and the next one is photosystem 1. Right? So that is the jet scheme, and then splitting of water also we talked. And now we are talking about cyclic and non-cyclic photophosphorylations, right? When we talk about, we are in the formation of high energy chemical intermediates where it can produce two ways. One is photophosphorylation and another one is chemoosmosis in chloroplast. What is photophosphorylation? Photophosphorylation is nothing but formation of ATP, phosphorylation, phosphorylating. What is actually addition of this phosphates? So this phosphate bond, when the phosphates forms bonds, the electrons are trapped inside this bond, and they are stored in the form of this bond. So when the bonds are broken, then it releases the energy. That's what, right? ATP converts to ADP to AMP. So and eventually ADP plus inorganic phosphate gives rise to ATP. So this occurs, right, in the presence of light and chlorophyll A. So this phosphorylation, addition of these phosphates, right, to the ADP would be taking place in the presence of light. So it is photophosphorylation. And ADP formation takes place to the following two steps. One is non-cyclic photophosphorylation, right, and the other one is cyclic photophosphorylation. So in the non-cyclic type of photophosphorylation, what you see is both ATP and NADPH2 are produced in this reaction. So in one of the photophosphorylation, you will not see this production of higher energy molecule, right? So what is the difference? Also, we'll try to see. Okay. Right. So if you have a textbook now, you can open and see this photophosphorylation, that is non-cyclic photophosphorylation, especially the jet scheme. When you see this jet scheme, during this non-cyclic photophosphorylation, the initial donor and final acceptors, right? Initially, there would be a donor of this electron, and there would be final acceptor electrons are actually different, right? So there is a difference between the initial acceptor and the donor, right? So electrons which are actually illuminated or hit this PS2 would be excited, and those electrons will be released and are passed to PS1 via some electron carriers, right? So when this PS1, that is electron, is finally provided to this provided to this pyridoxine in the sense like this, which actually eventually helps in the formation of NADPH2. So it is mean they are reducing. NADP is reduced to form NADPH2. So there is energy stored in the form of higher molecules, and this process would occur in a jet scheme. So there is a different electron, the light acceptor and light donor and acceptor are different when it comes to this non-cyclic photophosphorylation, right? So this production of NADPH2, especially the higher energy one, it is more higher energy than compared to ATP molecules, right? So that would be produced during this non-cyclic photophosphorylation through jet scheme. And then when it comes to cyclic photophosphorylation, in this process, the initial donor and the final acceptor of the electrons would be the same. So that's what it is cyclic, right? So initially there is a donor of the electron when the light hits, and this electrons would be donated and would be accepted. So both donor and acceptors would be the same. That would be chlorophyll A of PS1. So here only PS1 is involved in this photophosphorylation, right? So that you need to focus. So what is the difference between photophosphorylation and cyclic and non-cyclic? Where you will find that in this cyclic photophosphorylation, you will find only PS1 is involved, right? And when we talk about non-cyclic, you will find jet scheme, where you will find two photosystems would be involved, where donor and the acceptors would be the different. So these facts may come in the form of correct or incorrect statement. Okay, so the diagrams don't ignore. Okay, so the first the light hits this photosystem one, and then what happens is the electrons would get excited, and finally they reach to the primary acceptors, right? And then they send to this cytochrome complex, and then they would be storing the energy in the form of ATP, and again this few of the electrons after the few of the electrons which are not utilized by this ATP conversion would be again coming back to the original place. So this is cyclically happening actually. Okay. So when this cyclic photophosphorylation starts, so when here only PS1 is involved, right? So whenever the PS2 photosystem 2 is getting hampered or ceased their functions, so we are in the photosynthesis. We are talking about what we are actually talking about is mechanism of photosynthesis. We are talking, guys, right? A quick recap. So within the mechanism of photosynthesis, we have two things. One is one is photochemical phase, and other one is biosynthetic phase. So now even still now we are talking about only photosynthetic phase. Within the photosynthetic phase, we have four different events that occur. One is light absorption, splitting of water molecule, release of oxygen, formation of higher energy chemical intermediates. So we already talked about light absorption, and then splitting of the water molecule also we talked. And then when we talk about the formation of these higher energy molecules, especially we have two different types of reactions takes place. One is photophosphorylation and the chemoosmosis. Now we are there within the photophosphorylation. We have again two types. One is cyclic and non-cyclic. So and we start with non-cyclic, which has a jet stream which produces this NADPH2, and it will contains PS1 and PS2, and the donor of electrons and the acceptor of electrons are different, right? Isn't it? And in cyclic photophosphorylation, you will be finding both initial donor and the final acceptor of the electrons would be the same. So the same photosystem 1 or the chlorophyll A, which is actually donating, exciting these electrons or donating the electrons to the acceptor, and again it reaches to the final acceptor would be the same as the donor. So that's what it is cyclically happening. So in this only a PS1 is involved. So when this cyclic reaction takes place is whenever there is a cessation of photosynthesis 2 or non-cyclic photophosphorylation is stopped due to certain reasons. Okay. So is it clear? Is it clear, guys? Are you able to follow? So we are quickly going through the lesson contents actually. Okay, I'm explaining only few things which would be confusing. Okay, I can't explain entire lesson in 1 hour. Okay, or no one can really give a detailed explanation in 1 hour. Is it clear? Yeah. Okay, fine. So let's move on. So when we talk about the next type of this higher energy molecules formation, that is chemoosmosis in chloroplast. Like respiration in this photosynthesis to ADP synthesis linked to the development of proton gradient across the membrane. I told now, if you consider this is for example, okay, if you consider this book as membrane, this proton pump would be placed like this, right? So when the proton pump is placed like this, no? So the proton gradient would be, this is thylakoid membrane, right? So the proton, for example, we will take like this. So this proton gradient, for example, consider these guys as protons. Okay, these guys as protons. So these proton gradients would be produced inside the lumen, and they would be sent. While they're sending, electrons also passes, and finally what happens is this ATP synthase complex, that is F0 and F1 particles would be rotating like this, right? And when they are rotating, what happens is this ADP, for example, right? It is. Are you able to see? Okay. For example, right? So when this is rotating now within the membrane, what happens is, so when this is rotating like this, so this is F0 and F1 particles, and this is thylakoid membrane, right? So consider that this is inner membrane, so this is lumen, where these are the protons. So these protons, when they are transferring to this membrane, right, what happens is, let's take this. Okay, so the inorganic phosphate, this is inorganic phosphate and this is ADP, right? So during this rotation, what happens is it donates electrons, and this ADP would actually eventually become into ATP. So this is how the ATP synthase, which is present even in the mitochondria also. Okay, so that's what the you know, power generation plants, right? That is mitochondria also, you'll find this ATP synthase mechanism. And if you see this ATP synthase, I'll send a video. You try to see that after. You see this. Now quickly go to 20. Yeah, I'll send a link. Okay. Okay. So quickly go through the 2:22 minutes. Okay. So when you see this 2 minutes 22 seconds, you will find that this, you know, what I've just shown as an rough models, right? So F0 F1 particles would rotate. So where the proton gradient would be created, and then those proton, due to the proton gradient, then they will really produce this ATPs. Okay, you see later. Okay, so this is about the photochemical reactions, right? And then when we talk about biosynthetic phase, where we can call it as dark reaction or Blackman's reaction. Black dark. So Blackman is the one who actually observed this. So the biosynthetic phase. So in biosynthetic phase, which can be included, there are C3 pathways and C4 pathways, where you need to focus more on what is the difference between C3 and C4 pathways, right? And especially when we talk, these intermediates are important when we talk about this three different steps that occurs during this C3 pathway. Kelvin pathway, you have carboxylation, reduction, regeneration. So the balancing of the equations are very important, right? How many ATP are consumed, how many ATPs are utilized, how many NADPH are produced or consumed, that you need to know step by step. So at the end of the carboxylation, what are the products? What are the products? And then after the reduction, and what exactly regenerating, right? So this regeneration, for example, if you see this ribulose, right? So this ribulose, which is getting regenerated, right? What is the number of its carbon? So what is the initial substrate that actually involved in this carboxylation? So the CO2 is added to the carboxylated to form 3 PGA molecules, right? That is the first step within this Kelvin cycle. So these are the things that you need to focus, right? So you might have studied even 10th standard also, this photosynthesis and different steps, carboxylation, reduction, and regeneration especially, right? So try to see that who is actually the acceptor of the CO2. Who is, and if you really see that, during evolution also, we try to, so right, we try to, don't look that video completely right now. You can directly go to the 2.2, and you can see that how this proton pump is situated, right? And then how they are creating ATP molecules. Okay, come back, guys. So when we talk about this, what do you say, these pathways, right? So during the carboxylation and during the reduction and regeneration, you need to know the each product its carbon number also, right? So if you say this PGM molecule, phosphoglyceric acid, so you will find that the carbon number is 3. So there will be 3 member structures, 3 carbon member structures, right? So when you say this Rubisco, so this is an enzyme which is very abundant in the universe. So which enzyme is more abundant is that this Rubisco enzyme, where it has got both carboxylation activity as well as oxygenation activity, right? So that's what this Rubisco is more, because if you really go back to the the evolution of higher plants or evolution of this photosynthetic plant, you find the CO2 is too much abundant, where this enzyme is also abundant because it fixes the CO2. So this is one of the evolutionary aspect. At the same time, this is a protein you will find, or enzyme you will find, that abundant in the universe. So that might be one of the factual question, right? If Rubisco is there, that you can go ahead with that option, right? Right. So within this Kelvin cycle, you need to focus on different stages and the balancing. Okay, balancing of the equation, and how many ATP molecules are utilized and how many are produced. Also, you need to focus. So when we talk about C4, the Kranz anatomy, the mesophyll cells arrangement is important in Kranz anatomy. And what are the primary acceptors? If you see, of the CO2 is nothing but oxaloacetic acid. So OAA in the case of C4 plants. And if you see this C4 cycle, why the plants are actually really evolved in such a way, like why we have two different cycles, C4 and C3. That is where you need to more focus on, right? So when you talk about this C4 plant cycle, so in the C4 plant, both you will find this Kelvin cycle at the same time the C4 mechanism would occur. It means initially the CO2 is actually they've actually primary acceptors of the oxaloacetic acid, which is a four-membered carbon structure. That's what it is. C4. C3 is a three-membered that is PGA, 3 PGA molecules, that is a primary acceptor. But in the C4, the primary acceptors are OAA, that is oxaloacetic acid. And then oxaloacetic acid eventually converts, right, by utilizing the NADPH, they will be converted into malic acid, and then malic acid will be transported to the cells, which bundle sheath cells, right? So from this mesophyll cells you will find chloroplast, where initially in the C4 plants, this primary acceptor OA would accept this CO2 and eventually converts into malic acid, and that will be transported to the bundle sheath cells, where you will see Kelvin cycle would be occurring. So here what you need to understand is this pyruvate dehydrogen, pyruvate phosphate dikinase, or this step A, right, plus CO2, which would combine to form this OAA. So this pepA, which is formed from pyruvic acid, right, this conversion is important, right? So when there are no pyruvic acids, eventually this pyruvic acid is produced in the bundle sheath cells, right? So when the pyruvic acid is produced from the malic acid, right, and it would be releasing CO2. So that CO2 would be utilized for the Kelvin cycle. So it is, the CO2 is not directly fixed by this C4 plants, especially the Kelvin cycle, which is occurring in the C4 plant. So first the C4 cycle occurs, and then it is proceeded by C3 Kelvin cycle by splitting this malic acid into one CO2 and the pyruvic acid, that is 3 carbon structure. So this pyruvic acid again would be transported back into the mesophyll cells, and it would be combining with the CO2 by the enzyme, that is pep carboxylase. Here you need to focus on the enzymes which are involved in the first step of carbon fixation. Those are nothing but pep carboxylase or Rubisco. So these are the enzymes you need to focus. So you need to talk about this. What are the difference between C3, C4, and CAM crustacean acid metabolism? If you see this CAM pathways, right? So no. So what is the differences between these 3 pathways that you need to more focus on? Okay. So yeah. And coming to the photorespiration, in photorespiration, what are the different aspects, and what is the other name for this photorespiration? It is also called as glycolate metabolism, right? And if you see that what are the components that are required to undergo this photorespiration, especially you need chloroplast, peroxisomes, and mitochondria. These 3 structures together they involved in this photorespiration. So respiration is occurring in the presence of light, right? photorespiration, photons are fixed. How in the sense like the CO2 in the form of output reaches to the chloroplast and runs the Kelvin cycle. It means the CO2 required for the Kelvin cycle is actually again it is donated by this photorespiration. Okay, this photorespiration otherwise called as C2 cycle also. So we have C2, C3, C4 CAM. So this is how you need to write a tabular form, and you need to arrange all the components. And finally, factors affecting photosynthesis. Those graphs would be very much important, guys. Don't ignore them. So what exactly affects the photosynthesis? The internal factors and external factors. So there are some internal factors like protoplasmic factors, chlorophyll content, and byproducts accumulation, structure of the leaf. Whether they are, how much they are really, the leaf blade is exposed to the angle that which the leaf is exposed to the sunlight, or they are drooped like this. So those are the graphs that you need to focus. That is rate of photosynthesis affected by chlorophyll content, light intensity, or CO2 concentration. Clear? So here we'll be talking about limiting factors, right? Why is that? Because limiting factor is one of the important factors here. Photosynthesis may get affected by different aspects, but only any one of the component would be really determining the entire reaction. The minimal amount change in that, right? The minimum amount of change that occurs within that component would really hamper the entire reaction. For example, CO2 you take. For example, you have light intensity, very good. You have leaf blade arrangement, very good. You have humidity, you have external, internal temperature, water, everything is fine. But the CO2 is very low concentration. For example, you need 0.05% of CO2 to really fix this normal photosynthesis. But if you have 0.02%, obviously the photosynthesis will not be occurring. So that is a limiting factor. Until the limiting factor is provided, the photosynthesis will not be carried out. So what happens if you consider if you consider the rate of reaction, right? So what happens is the photosynthesis would not take place. So this is a rate at which rate of photosynthesis, and if you consider the CO2 concentration, right? Until we provide certain CO2 concentration, what happens is you will not be having that photosynthesis to take place. But at the same time, if you provide more CO2 also, some other component will be turning to be a limiting factor. So these factors are concerned internal and both external factors, and limiting factors or that you need to more focus on. Is that clear? So law of limiting factors are very important in the case of this photosynthesis. So these are the important aspects you need to follow. So especially the dark, the bolded letters and the concept around it is what you need to focus. PEP carboxylase, enzymes and their functions and locations also very much important. And differences between C3, C2, C3, C4 and CAM plants that you need to focus. Is that clear? So this is about the lesson where you need to more focus on the limiting factors also without any sort of ignorance. Like you shouldn't be ignoring any topic in this physiology chapters, right? So with this we completed this lesson, and tomorrow we'll try to we'll have body fluids, right? So if you complete this lesson, stay in this lesson and at least read for at least 2 to 3 hours, right? 1 hour we can cover the entire outline. Means you can even in depth you can study 2 to 3 hours is more than enough. Okay. Okay then. Then see you in the next class. Take care. Stay home. Stay safe. Thank you guys for joining in the live session. Thank you.